So, hello everyone. My name is Gilad. I used to be part of the advanced analytics team here in Israel, and then I relocated to Portland, Oregon, where I manage the project that I'm going to talk to you about. So, cancer. Cancer is something that has touched, as Karen said, all of us. Recent studies have showed that 50% of the population will have the risk of getting cancer in their lifetime, of people born after 1960. So just think about it. One of the people sitting next to you will have this terrible disease. A little bit of what's new and what's happening in genomics before we go into the technologies is this is a graph of Moore's law, which is the white line that you can see there, how we're increasing the speed of the CPUs while reducing the cost. And next to it, the green line with the dots is the cost of sequencing. You can see how the cost of sequencing has dropped dramatically from the first person that we sequenced in 2001 that cost over $100 million. Today we're close to $1,500 for sequencing one person. Of course, for cancer, we need to sequence a person twice because we have his healthy tumor and the cancerous tumor, cell, but it's still, the cost has dropped dramatically. And obviously, when cost of things drop, we see much more data being generated, right? So in the beginning, we saw a very vertical line of sequencing. It just cost too much. We had minimal amount of sequencing going on. But as the prices drop, the amount of data that is being generated grows dramatically. In the beginning, it was all government-sponsored projects that generated all these sequence data because they're the only ones that had still the amounts to generate the data. But as the cost drops, we see private people and different academic institutes generating the data. So I'm not a biologist or genomics. I'm a technical person, but I want to explain a little bit what does sequencing mean, and what does the flow of doing precision medicine, of doing real treatment based on our genomics and imaging data. So everything starts with an appliance that starts in the left side of the slide, where we take a tissue of a person, we put it in this appliance. There's a lot of chemistry and biology going on there to actually start generating the data on the DNA of a person. This process takes around a day to 4 days, depend on the depth you want to do. And Intel is helping the appliance providers to accelerate that work as well. But what comes out of this is actually not a DNA of a person. What comes out of this is pieces of a puzzle. Okay, so it's pieces of our DNA, billions and billions of pieces of our DNA that then need to be assembled. Then we need to build that puzzle into our own sequence data and our own genomics data. That is a highly complex compute task. A lot of analytics involves a lot of algorithms in order to build that sequence. That sequencing build takes hundreds of hours to generate, and in the end, we get a sequenced person DNA. Once we have that, then we start doing large population analytics, or GWAS, wide association studies, where we take multiple patients, multiple cancer tissues and DNAs, and generate everything together and start finding interesting insights over that data. And then we actually start building models to build predictions over what drug actually works for what mutation and so on. So as you can see, this process is very, very long. It takes weeks and months to actually generate. And actually, we don't know enough. We don't know enough about what mutations cause what. So a little bit more about the data itself. So when we sequence one person, it actually generates 1 terabyte of data. It depends on the parameters and all sorts of different aspects, but it's about 1 terabyte of data per one person. According to the American Association of Cancer Research, in 2015 only, we are going to get 1.65 million new patients in the US only. If we sequence all of them, we generate 4 exabytes of data. This is only the US, this is only 2015. So the data is actually huge. So if we have so much data, what's holding us back from finding the insight, right? We have a lot of data. This whole conference is, there's great data, there's big data, let's get all the information out of it, let's find the insights, and we'll find a cure for this disease. So it turns out that only 4% of the data is actually accessible for research, is actually available for the public to start doing analytics and doing analysis on this data. And the reason the 96% are below the surface and stored in is that it's stored in different silos in different academic institutes. All these institutes that sequence, they get a grant, they get a few $1 million, they sequence a group of patients, and then they have all the data located in their own data center, whether it's on-prem or in the cloud, doesn't matter, but it's their environment that is totally controlled by them, and they're not letting anyone else access that data. There are 2 main reasons why they're not letting others access the data. One is, as we said, the data is huge. Start moving around all this data for every researcher in every point of the world that wants to do the analysis just doesn't make sense, it doesn't scale. And the other, which is the main issue, is they want to have control over the data. This data is their IP. There are all sorts of law issues around moving it. Genomics data is the most private data there is, so there are all sorts of privacy constraints. So they want to keep tight control over the data and not just put it on Amazon, Google, or any one of the clouds for anyone to consume. So this brought us an intel to the idea that there needs to be a different way. There needs to be a way for everyone to collaborate over the data, but still leave ownership of the data to the data owner. And that's the collaborative analytics that we want to enable with this platform. So we have designed a platform. It has 4 main technology pillars, which I'll go into details in a sec. The first kind of idea is, as I said, to have each of the data owners hold the data local and enable a federation of data owners to interact between them. The second is we have to make things run faster. We have to do all sorts of accelerations, leveraging Intel architecture for things to run much, much faster than what they currently do. The third one is security, and that's enable a new security paradigm to do joint computation but remain privacy preservation, and I'll explain that. And the last one is enable this to scale. So most of the analytics in this domain is done on files. That obviously doesn't scale, that obviously doesn't run fast enough, and we're enabling a new database that can actually run these things faster. So the first thing is the federation. So this is a map of the US. You can't really see it, but there are numbers next to each state representing the amount of new patients in 2015 in each of these states. new patients having cancer in each of these states. Obviously, you can see that it makes sense. The data is distributed between all the states. Everyone has cancer. And the data and sequencing is being generated in each of the states. So this relates to the fact that the data is huge and the data is spread out everywhere, and it doesn't make sense for us to move this data around. So in order, I'll do a short animation, and in order to solve this problem, we've created a platform with 3 different institutes that each have their own data. These are real 3 academic cancer institutes. OHSU in Oregon, OICR is the Toronto in Canada, and Dana-Farber is in Boston. They each have their own data sets locally, genomics data, but they want to do joint analytics between them, right? They want to leverage the data that each other has in order to find more insights and find the kind of the long tail of the cancer, because cancer is not just one disease. Cancer is actually hundreds and thousands, and we don't know actually how many diseases it actually is. So what we built is a central function that receives some kind of a DSL, a domain-specific language. We call it WDL or WIDO. It stands for Workflow Definition Language. It's not something that we've created. We've worked with the industry, mainly the Broad Institute of MIT and Harvard, which are leaders in this field, to define this language. And this actually, a user submits a workflow to be constructed in this environment. Once he submits it, there's a workflow engine in the central function that understands where the data sets that the user actually wants to run his analysis on and distributes the compute to the data, right? Instead of aggregating all the data to one cloud, we're leaving the data at rest where it was generated and sending the compute through standard processors of Linux containers, Docker images, to run on the data at where it resides. Once that compute runs, and these are intense compute, batch computes that run hours to understand, to bring insights to the user, there's a fetch that actually aggregates all the data into the central function, does some function to reduce, and then reports all the results to the user. So you can think about this as a giant MapReduce, right? Where the map function actually runs at the different institutes, and then the reduce function runs on the central function and returns the result to the user. So that's how we actually federate. There's, of course, a lot of details behind it, and in the end of the slide, there's a lot of links to GitHub repositories. These are all open source projects that everyone can go and look at. The performance issues, again, we said these things take long. In the bars here, you can see the amount of million server hours that it takes to do the same compute, sequencing compute on each of the 2015 new data sets. In the little bar, you can see the millions of dollars the power just cost us. So even the power costs $53 million to sequence all these patients, only the new 2015 patients. So we have to make this faster. And we're working, again, Intel's not developing the analytics for this. Intel is working with the best-in-class analytic providers, such as Broad, to accelerate working with accelerations from existing Intel hardware, such as AVX, leveraging FPGAs, rewriting GATK, which is the Genomic Analytics Toolkit, which is the most commonly used analytics tool for genomic, into Spark. Intel is a co-author of GATK in Spark, that we're helping this to actually scale, to run faster, and to run better on Intel architecture. On top of that, there's the privacy. So the MapReduce paradigm works great for some analytics. But think about if you want to calculate a median of a set of numbers, you have to have all the data co-located. You sort the data, then find the median of a set of numbers. In order to help with that, we've developed a new capability that enables a software stack from hardware to the software, from the hypervisor, OS, and applications that enable you to actually secure the data and let the data leave your premise, but ensure the data is not breached at any time. No one sees your raw data, not a system administrator, not a third party. No one can actually see it except the application that you pre-approved to run on your data. So we're running a little bit late, so I won't have time to go through the entire animation of this, but the idea is that the data leaves encrypted the premise of the user. It stays encrypted when it's stored in a secured environment, and it's only decrypted by the application that computes the data to it. No one sees that. There's no kind of an Edward Snowden or any administrator that could actually sniff the data at runtime. And it's only available for the application that the user approved. Then the result sends back to the user, and the data is deleted. Last thing is a new database. As I said, usually the community today uses files to do the major wide association studies. This doesn't scale. And what we found out was that genomics data is really a very, very sparse matrix. So we're all the same in 99.999% of our genome. And we're only concerned about the differences. We're only concerned about the variances and the mutations that each one of us has. So you can think about a matrix where the rows are samples and the columns are positions in the genome. So we have 3 billion positions in the genome. And we have values only where each individual has a mutation. So that generates a very, very sparse matrix. Together with the Intel Science and Technology Center in MIT, we'll develop a new database called TileDB. On top of this TileDB database, we've developed a layer that is called GenomicsDB, which is specific for genomics. And what this enables us to scale, enables us to run much faster, both in reducing the amount of disk that is needed, plus the performance that it runs, which we've already deployed this in production environments, and it runs over 100x scale faster than existing files. Again, this is a complete open source that we want as many people to use as possible. So to conclude, these are our 4 kind of pillars of what we wanted to do in this platform. We wanted to move the analytics to the data, accelerate the analytics, make the analytics secure, and when you have to move the data, secure the data, plus enable this to scale. 2 examples of the new kind of, once we have this application up and running, which we have, and start the things we're starting to see around machine learning that we're tackling now is how do you actually do federated analytics? How do you aggregate different models that you built in different sites, and you want to aggregate everything to one single model? And the second is adding the imaging data into the genomics data. Imaging brings in a whole new aspect of challenges. The data we have problems in getting classified data. It's much easier to classify dogs and cats and things like that. But to classify a specific cancer cell, what exactly type you need, very highly sophisticated people to actually do this. There's a lot of context that is needed on top of the image to do all the analysis. And we're not dealing only with two-dimensional images. We're dealing with many three-dimensional and multi-data sets. So Intel has kind of given a challenge to itself and to the industry that we want to enable all in a day. That's kind of our vision. The platform that I just explained is the first step towards this vision, where we want a patient to come to his doctor and get sequenced, get image analysis, get all the information that is available about this patient, and provide him a real precision medicine, real personalized treatment for the user, all in one day. Meaning one day he'll come to the doctor, get sequenced, get imaged, run the models, and get the treatment that he actually wants to get, that he should get based on his personal data. And our goal is to enable this and for the industry to enable this by 2020. And that's it. Thank you very much.